Can you see the screen?
Yes.
Because I do not have any return from the screen you have. And here it's okay?
Yeah, full screen mode.
So it's okay.
Yes. Yes.
So today I will speak about Lactococcus lactis that has been revealed the last 20 years to be an attractive cell factory for the expression of membrane protein. What are membrane protein? They are encoded by 1/3 of the human gene, involved in many important cellular processes, and are therefore the target of more than 70% of the chemical drug available on the market for the moment. But there are still some limits for the structural and functional studies. First, they are present in low amount, naturally in the cells. They present a high hydrophobicity, and there is many difficulties to express them under the functional state in different heterologous systems, because sometimes there is toxicity of some recombinant protein. They can be also expressed at very low expression level, and they can be also misfolding of the protein produced. That's why there is only 500, almost 500 3D structure of membrane protein if you compare to more than 100,000 of structure available for the soluble protein up to now. So what is this Lactococcus lactis? It's a gram-positive bacteria that is generally regarded as safe by the FDA. And it belongs to the group of probiotic by the European group. It is used mostly in agro-industry and biotechnology for production of fermented food. It has been intensively studied at the level of physiology genetic genomics, and the genome is sequenced for many different strains, but only from 10 years ago. It can be also used to produce heterologous protein, such as antigen and therapeutic protein delivery that can be used in medicine, but also for biotechnology for production of soluble and membrane protein. The promoter it's an expression based on promoter most of the time, and many strategy has been used in order to improve the gene cloning that could be a little bit difficult with this bacteria. Most of the time, if you use a classical cloning of gene, you need to clone the gene between the NCO at the 5' terminal of your gene. That is an obligatory site, and another site of the multi-cloning site at the C terminal of the gene. This multi-cloning site is very small, only less than 10 sites for restriction enzyme. That's why new strategy has been developed for the 15 years, either belonging to or based on ligation-independent cloning and vector backbone exchange by Pullman and collaborator. We developed a strategy that was compatible with gateway vector 10 years ago. All other vector has been developed also by another group by Duya et al. 10 years ago, that are containing gateway cassette for expression of soluble protein. And only a few years ago, another group by Berluck is developing vectors that can be used in order to express protein or membrane protein at the same time as another protein. Other strategy has been developed in order to enhance the protein expression within the bacteria by improving the strain and by fusing GFP and antibiotic resistant gene. Another strain has also been developed, the strain M4, almost 10 years ago. And this strain is helping and try to overcome the problem of plasmid instability within the bacteria. So when trying to express membrane protein in Lactococcus lactis, most of the time it's the NIS system that is used for nisin-induced control gene expression. This NIS system has been used for many different things, as you can see here on the right, for the vaccine delivery to induce immune response, at the industrial scale application, but also for cloning of toxic gene, for expression and production of cytokine, for protein secretion, for production of bacteriocin, expression of homologous gene, but also of heterologous gene from bacteria. And what is interesting me since 15 years now, it's the expression of membrane protein from both prokaryotic and eukaryotic origin. So how it works? First, nisin met and go and fixed on its own receptor on the membrane. Then there is a transduction of the signal until the plasmid that is containing the gene of the protein of interest placed under the control of the inductible promoter that is kinase A. Then there is expression of the proteins that can be either soluble or membrane, or also secreted outside the bacteria. And this nisin is a 34 amino acid polypeptide antibiotic that allow induction of membrane protein and other protein with sub-inhibitory concentration. So in the lab, what we process for the moment, we use different strains obtained from the company NISO. The first one is the 9700 that is producing nisin, and the 9000 that is the strain that is a host strain for the nisin-inducible vector. Most of the time for membrane protein, we are using this PNZ 8148, that is the vector with the chloramphenicol gene, this promoter inductible by nisin, and the multi-cloning site I spoke before with this NCO1 site that is obligatory for translational fusion. So now how we can extract and we can isolate membrane protein with this bacteria. We culture them in the M17 medium in presence of glucose and in the right antibiotic, most of the time chloramphenicol. In shot bottle directly because it's an anaerobic, aerobic facultative bacteria. And we induce at optical density close to 0.8. Then we let the bacteria grow for 4 more hours at 30 degrees. The induction is performed by a homemade nisin that is contained in the supermanatan from the bacteria NZ 9700. Then in order to recover and to have the membrane protein, the bacteria are released with a disruptor from the satiety constant at 3 times the atmospheric pressure. The first centrifugation at 10,000 G allow to remove the intact bacteria and the cell wall, and a further ultracentrifugation at 150,000 G allow to concentrate membrane protein and to remove the soluble one. We obtain yield from 60 milligrams of total membrane protein per liter culture. These bacteria present many different advantages over the most and the famous Escherichia coli bacteria. Then you have a tightly regulated system that is the NIS system that allows the expression of toxic gene product. You have also a functional expression and a right localization of your protein that are soluble on the membrane, and we can say that is without adding peptide leader sequence to this protein. It possesses only one glycolipid-rich membrane that allows to integrate functional protein and to perform functional studies, either at the whole cell level or within membrane vesicles. You can easily label the protein because it possesses an auxotrophic metabolism, and you can incorporate selenomethionine. It do not possess extracellular protease, and it grows under fermentation or respiration. And the most important advantage over E. coli is that there is no formation of inclusion bodies. You can use this bacteria for production of protein for food, feed, and pharmaceutical use, either oral or topical use. You can use for production of ingredient, amino acid, polysaccharide, and flavor. You can have an in situ production within the body, and it's a cofactor independent biocatalysis. So up to now, there is 115 membrane proteins that has been expressed in this bacteria since 20 years. Some of them are from prokaryotic origin from the bacteria themselves, 23, and then half are from prokaryotic origin and the other half from eukaryotic origin, either from yeast, from plant, or from human. This bacteria allow to express protein, membrane protein from peripheral to protein that possess 14 transmembrane helices from 12 to 150 kilodaltons of size. And as you can see on the right side over here, there is two main clusters of proteins that has been expressed either close to 6 transmembrane domain or 12 transmembrane domain. All this membrane protein has been expressed under the native functional oligomeric state. Mainly transporter has been expressed in this bacteria. Some also in blue here, the mitochondrial protein has been expressed that were originate at the beginning from Saccharomyces cerevisiae, and the expression is linked to the bacterial lipid composition in terms of yield. From now, I want to focus on some special membrane proteins that we were studying using this bacteria in order to go further in the functional study of the protein themselves. First, we have used the bacteria in order to express the chloroplast ATP-AD pitone sulcator. It's a large membrane protein of 60 kilodalton, and that possesses a chloroplast transit sequence that we do not truncate it. It's present in plant and in bacteria, and it exchanges ATP against ADP and PI. PI is the co-substrate here. And then it performs the ATP import into chloroplast. You will see in the next slide AAC that performs the same things in mitochondria. So even produced at 0.2 total membrane protein, this protein was still active on transport ATP. As you can see here, the small black band corresponding to the protein on the western blot. And we were able, using radioactive ATP, to measure the function of the protein. I was spoken about these mitochondrial carriers that was mostly expressed in the bacteria. They have 6 transmembrane domain, the size of 30 kilodalton, mostly exclusively from eukaryotic origin. And they can transport different substrates such as nucleotide and pyruvate. In Lactococcus, they are expressed at a level from 1 to 10% of total membrane protein under their monomer or heterodimer form. And when performing a functional test in Lactococcus, this functional test can be either performed on the wall cell, on the bacteria themselves, but also on proteolaprosome after solubilization, purification, and reconstitution, using different substrates that can be or not radioactive. And as you can see here, for the 5 different membrane proteins, they are still active. I wanted to focus this slide on the human mitochondrial AAC1, that is also a mitochondrial membrane protein of 6 helices of 30 kilodalton, and that is expressed in Lactococcus at a level of 1% of total membrane protein, as you can see here on this comestible staining. And we performed functional tests in the bacteria, and we were able to have a look and to determine the ADP uptake by the bacteria that contains the membrane protein of interest. Then, and for the last part of the topic, I wanted to focus on ABC transporter, since this bacteria is the right bacteria, is the appropriate one if you want to characterize or to perform functional studies on this ABC transporter. ABC transporter belong to different family from A to G, and they can be hard for full transporter, homo or heterodimeric protein. They contain ATP binding cassette, either 1 or 2 of these cassettes containing the WOCA A, WOCA B, and the ABC signature, as you can see over here in the nucleotide binding domain. And this cassette is performing the ATP binding and hydrolysis. They are present in all the studied organisms, and many different types of transport system has been described for them. For ABCB and ABCC, they are responsible for drug resistant and involved in catabolite and in toxic xenobiotic in mammals' livers. And in humans, there are 2 well-known ABC transporters, the CFTR and the ionic channel. If you want to perform a functional characterization of this ABC transporter, you have many different studies that has been performed on the LMRA from Lactococcus lactis, the MSBA and SAV 1866 from E. coli, the human ABCG2, and the heterodimer TM287, 288. One most advantage of Lactococcus is there is a strain that is delayed of the 3 main homologues of the ABC transporter LMR A, C, and D, and that help in order to measure transport and ATPase activity and ATP binding. Lactococcus allow to reconstitute also homo or heterodimer after co-expression of protein. That is a big advantage of E. coli for functional characterization of such membrane protein. And then as an example, I will speak about this heterodimer TM287, 288, and for which the crystallization has been performed with proteins that were produced in Escherichia coli, but when they perform the functional characterization of this heterodimer, it has been performed in Lactococcus lactis that has been delayed of this PGP homologue LMA A, C, and D, as you can see over here on the right part of the slide. Then to speak about this 3D structure, Lactococcus lactis start to be an expression system for the determination of such structure of membrane protein. Up to now, 20 of this 3D structure has been resolved in the 10 last years, mostly of ABC transporter, secondary transporter, but also the ATP MGPI carrier. And as a conclusion, as to summarize this presentation, Lactococcus lactis is an alternative and interesting host for expression of protein, including membrane protein, most of the time of pharmaceutical interest. The NIS system allows a tightly regulated expression of this protein, and the bacteria help and is a nice and appropriate bacteria for functional and structural characterization of protein. This protein can be used either in therapeutic for specific protein expression in probiotic, but also for many other diverse purposes, such as device development, because I'm belonging to an engineer science institute. Thank you for your attention. And I would like to acknowledge 3 different labs where I was studying on this bacteria: the CEA at Grenoble in France, the CEA close to Paris at Saclay, and the institute I'm joining 7 years ago now at Besançon, the Institute FEMTO ST. Thank you for your attention.